Hi everyone, welcome to S&S Bioinfo. Today's topic is Gregor Mendel and his experiments. Gregor Johann Mendel was a monk who lived in Austria in 19th century. He was also a natural science teacher. He is known as the father of genetics as he has given basic ideas of the principles of inheritance of traits. There were a lot of garden pea plants in his monastery. He was very much attracted in doing experiments with those plants. He conducted several hybridization experiments to study the inheritance of genes in pea plants. Let's learn more about his experiments. I told you that he conducted several hybridization experiments with the garden pea. The scientific name of garden pea plant is Pisum sativum. The scientific name of garden pea is Pisum sativum. Then he selected 7 characters and 7 pairs of contrasting traits to study the inheritance of characters in garden pea plants. Before we study about 7 characters and 7 pairs of contrasting traits, you must know what's the difference between character and a trait. Character is a distinct morphological and physiological feature of an individual. For example, the color of my hair. Color is a character, while the trait is the expression of a character. The color of the hair can be different in different person. Black hair, grey hair, brown hair, and so on. So those are known as traits. Contrasting traits means opposing traits. For example, tall, dwarf. Tall is the one trait, dwarf is the opposing trait. Gregor Mendel selected garden pea for his experiment because of some reasons. First one, there were wide varieties of pea plants. It was easy to cultivate. It was easily available in the locality. The flowers are bisexual, so it was easy to cross-pollinate. The plant is short-term or annual plant, thus he could produce several varieties within short period of time. He could make many results and he could study about the inheritance of genes. Now let's study the characters of pea plants and 7 pairs of contrasting traits. The 7 characters are, first of all I hear characters. Number 1, stem height. Stem height. Second character, shape of the pod. Shape of the pod. Third, color of the pod. Color of the pod. Then shape of the seed. Shape of the seed. Fifth one, color of the seed. Color of the seed. Position of the flower. Then color of the flower. Color of the flower. So these are the 7 characters selected by Mendel for his hybridization experiments. The 7 pairs of contrasting traits are, let us categorize them as dominant trait and recessive trait. Recessive trait. These two are opposing traits. So for stem height, the dominant trait is tallness. Here it is dwarfness. Dwarfness. Then shape of the pod. Shape of the pod, inflated, dominant, constricted, recessive. Then color of the pod, green, yellow. Then shape of the seed, round, wrinkled. Then color of the seed, yellow, green. Position of the flower, axial and terminal. Color of the flower. Violet or purple, then white. These were the 7 pairs of contrasting traits selected by Mendel to study the inheritance of one gene and 2 genes. So tallness, inflated, green, round, yellow, axial, violet. All these are dominant traits. While dwarfness, constricted shape, yellow color, wrinkled, green, terminal, white are recessive traits. Five steps were involved in his experiments. They are: number 1, selection of plants. Number 2, hybridization. 3, self-pollination. Number 4, calculations. Number 5, collection of seeds. First of all, selection of plants. Gregor Mendel selected pure varieties or true breeding lines of garden pea for his experiment. True breeding lines. Do you know what is true breeding lines or pure variety? Pure variety can be produced by repeated self-pollination. By repeated self-pollination, the parental traits can be retained in the successive generation. So, such type of trait is called a pure trait. So, the pure trait is controlled by homozygous pair of allele. For example, homozygous dominant. This is a pure variety, pure tall. Then this is a homozygous recessive allele, a pair of recessive allele. So, pure dwarf. The next step is hybridization. Hybridization is the process of crossing of 2 genetically dissimilar parents to produce a new variety or a hybrid. Plants with the contrasting traits were cross-pollinated artificially. And what is cross-pollination? Transfer of pollen grains from the anther of one flower to the stigma of another flower, which are genetically different. He conducted cross-pollination in the garden pea plants. Let's learn the steps that are involved in the cross-pollination in Mendelian experiments. They are, first of all, emasculation. Second one, bagging. Third one, dusting. Fourth one, tagging. Fifth one, crosses. Finally, collection of seeds. Emasculation, bagging, dusting, tagging, crosses and collection of the seeds were the steps involved in cross-pollination. The first step is emasculation. Emasculation is the process of removal of anther from the flower. You know that garden pea is a bisexual plant. The flowers are bisexual in nature. Bisexual flower consists of both male and female reproductive organs. By removing the anther from the bisexual flower, Mendel made them as a female flower. Thus he made male parents and female parents to do cross-pollination. The next step is bagging. The flowers were covered with the paper bags from the beginning. It was open only at the time of emasculation and dusting. The next one is dusting. Dusting is the process of transfer of pollen grain from the anther of one flower to the stigma of another flower. It was done by using a fine brush. The next step is tagging. Separate tags were attached to the plants to indicate the crosses. Next one, crosses. Separate crosses were performed to study the inheritance of one gene and two genes. There were monohybrid crosses and dihybrid crosses. Finally, collection of seeds. Seeds were collected separately. Those seeds are known as F1 seeds or first generation seeds. Those seeds are known as F1 seeds or first generation seeds or first filial seeds. These seeds are sown to the next season. Those plants are known as hybrid plants. Hybrid plants. Thus, Gregor Mendel could produce several hybrid plants by hybridization technique. Actually, his experiment involves cross-pollination and self-pollination. Those are the two phases in his experiment. Cross-pollination over. Thereafter, he allowed the hybrid plant to undergo self-pollination. And after self-pollination, calculations were made separately, and those calculations were according to the law of probability in mathematics. Finally, collection of seeds. Seeds were collected separately, and those seeds are known as F2 seeds. F2 seeds or second filial seeds. That means second generation seeds. Thus, Gregor Mendel conducted several monohybrid crosses and dihybrid crosses to study the inheritance of genes. Based on his observation, he proposed the principles of inheritance. We will learn about the principles of inheritance of Gregor Mendel in the upcoming classes. Now, I wind up this session. Thank you and have nice moments.